Hi, I'm Professor Jiaqin Zhang from National Yangming Jiaotong University, Taiwan. On behalf of our international research team, it's my pleasure to introduce a novel sensing platform for the detection of SARS-CoV-2 virus and for screening of the antivirus infectious disease drugs. This research team is quite unique. We are from different universities in Taiwan and Malaysia. Although we could not work together side by side in this pandemic era, we managed to work cooperatively to solve problems and exchange ideas. It turned out works. In the following talk, I will show you how this sensing platform was developed and what this sensing platform can do. The COVID-19 pandemic is a global health crisis that has a deep impact on our world and our daily lives. More than 220 million people infected and 4.5 million people dead due to this newly emerging infectious disease. Furthermore, a lot of variants of this virus have been identified in different areas. Unfortunately, the spreading of these variants, especially the Delta variant, is much faster than that of wild type. Some breakthrough infections are also observed in the fully vaccinated people. Therefore, a detection of SARS-CoV-2 and its neutralized antibody is important for better controlling the spread of this newly emerging infectious disease. In this study, we first identify and isolate novel molecules that can specifically recognize and binding SARS-CoV-2. With the cooperation of our industrial partner, we have developed a palladium-thin-film-based flexible electrochemical electrode, which can be used to conjugate biomolecules directly without any matrix. The developed electrode can detect the virus within 15 to 21 minutes in a sample as low as 1 microliter. The amount of drug can be detected is as low as 4 micrograms during drug screening. Compared to vehicle SPR, the amount of sensing molecule, which to functionalize the sensing probe, is 1 of 10,000 of the SPR. The sensing sample only requires 1 of 100 or to 1 of 30 of the SPR. Therefore, our sensing platform has an advantage over conventional approaches. As shown in this slide, an approximate 20 nanometers palladium nano-thin-film has been developed, and the electrode possesses the excellent electrical property, which is similar to that conventional gold electrode. In addition, a USB-type adapter was developed to connect the generated palladium nano-thin-film electrode for simultaneously multi-sample detection. Interestingly, the palladium nano-thin-film has a unique structure that can induce local surface plasmon resonance and enhance surface electric field. This phenomenon allows the Raman signal of protein sample as low as 0.8 microgram of palladium nano-thin-film can be detected. By using palladium nano-thin-film, a SARS-CoV-2 sensor can be generated by functionalizing the electrode surface with ACE2. Interestingly, we found that not only SARS-CoV-2 virus, but the SG614G variant can also be detected with higher affinity than that of wild-type strain. In addition, our palladium nano-thin-film electrochemical sensor can detect other variants too. The SARS-CoV-2 virus infects the host cell by binding to the ACE2. Therefore, the drug that interferes with the binding of virus to ACE2 can be screened by using this biosensing system. We found 4 potential leads, such as the ramipril and the ramipril, which can suppress virus binding from 72% to 42%. Surprisingly, we also found some existing drugs which can enhance the binding affinity from 25 to 174-fold. Similar trend can be found in cell model by quantitative PCR. This result confirmed the study on our biosensing platform. By using our biosensing platform, the breast cancer surface marker HER2 can be specifically detected within 15 minutes with a lowest detection limit of 0.1 nanogram per ml. Furthermore, our biosensing platform can be functionalized with specific DNA, which can be used to screen the DNA sequence-specific anti-cancer drug. In this slide, we demonstrate that M4N, which can bind SP1 DNA specifically. However, doxorubicin can bind both SP1 and the non-SP1 DNA. Taxol did not bind with DNA molecules. In summary, we have developed a rapid and sensing biosensing platform for identifying SARS-CoV-2, breast cancer, and antivirus infection drug or anti-cancer drug. We have screened and identified 4 antivirus infectious drugs by using the developed biosensing platform. The developed biosensing platform may be further adapted for the identification of drugs against other types of infectious disease and cancer. This discovery has been published in world-leading international journals, and the 2 U.S. provision patents have been filed. This research is a joint effort between the National Yanmenjiaotong University, Chang'an University Epidemic Prevention Center, National Chengkong University Epidemic Prevention Center, and the University of Malaya Faculty of Medicine and the Faculty of Pharmacy. This technique development has been supported by Ministry of Science and Technology, Taiwan, and the Ministry of Education, Taiwan. Here is my contact information. You are welcome to contact me for any cooperation issue. Thanks for your attention.